Hello and welcome back to the Cheeky Science Show. So today we're going to address the question of what are the primary causes of aging. So last time I introduced you to the hallmarks of aging, for which there are 9 different hallmarks, and these different hallmarks are categorized into 3 different components: the primary causes of aging, the antagonistic hallmarks, and the systemic or integrative hallmarks. In this video, we'll just look at the primary causes of aging. The first of which is DNA or genetic instability. But how does this actually contribute to aging? So to understand the involvement of DNA instability in aging, evidence that supports both increased damage and advanced aging, and evidence showing that decreasing the damage delays aging, are needed. So evidence for the former of these comes from both artificial induction of damage and increased aging, and from genetic diseases such as progeria, where increased damage advances the aging syndrome. Other cases where we know that DNA mutations, a form of DNA damage, are important for disease progression are seen in cancer and neurodegeneration, both as age-associated diseases. So how do you even get DNA instability? So this can be split into 2 categories, one of which is intrinsic or endogenous damage. The other is extrinsic or exogenous damage. So the intrinsic or the endogenous sources of damage include reactive oxygen species, spontaneous damage, and also mutations that can occur from DNA replication errors. The extrinsic or exogenous sources of damage include UV radiation and chemical exposure, such as carcinogens. And so, using one of our favourite model organisms, the mouse, many genetic models have been created to show the role of DNA instability in contributing to aging. For example, models with mutations in genes for p53, guanine, and other helicases, which are genes involved in the recognition, response, and repair of DNA damage, cause an accelerated aging phenotype in the mice. In addition, the same thing has been seen with mutations in the protein that encodes the polymerase that replicates the mitochondria DNA, have also shown an accelerated aging phenotype in the mouse. So all of these studies really demonstrate the role of genetic instability in contributing to aging. But is this just a correlation, or is it actually a causation? To really get an understanding of this, we really need to have models whereby we can prevent damage and see a reduced aging rate. So the next primary cause of aging is loss of proteostasis. And so proteostasis takes into account the synthesis, folding, and degradation of a protein. And so for proteins to be functional, they need to be correctly folded. Hence, loss of proteostasis results in a loss of function, and this contributes to aging. So what actually happens during aging? So as you age, there is generally seen a reduced protein synthesis rate, but also a reduced degradation rate of protein. So this means that the actual total level of protein in a cell remains the same. However, that same protein has been present in the cell for a longer time period, and during that time in the cell, it could accumulate modifications or become unfolded, and hence it loses its function. So whilst the amount of protein may be the same, the protein's ability to function correctly is reduced. And so another consequence of a protein being in the cell for a long period of time is that it's more likely to aggregate, and aggregation of protein is very key in aging phenotypes. For example, neurodegeneration and Alzheimer's disease, you have aggregation of amyloid beta, and this aggregation is thought to be the key trigger in the development of Alzheimer's disease. Evidence for the support of loss of proteostasis in aging has also come from another really famous model organism, the worm, or C. elegans to be more precise. And so a study was conducted whereby they took all the protein from either a young or an old C. elegans, and what they saw was that there was an increased insoluble proportion of protein at the later stage of their life, and insoluble protein arises through aggregation. Hence, you have protein aggregation seems to be correlating, whether or not it's causing, but at least correlating with the aging phenotype. The next primary cause of aging is telomere attrition. So telomeres are repetitive DNA sequences that are found at the end of chromosomes, which are just large segments of DNA. And so they kind of act like shoe caps that retain the integrity of the chromosome. However, as you age, these telomeres shorten over time, and so this integrity gets lost. This loss of integrity could result in tumorigenesis. Therefore, the cell has mechanisms to kind of cope with the loss of telomeres. This mechanism is referred to as cellular senescence, or replicative senescence in this case. And so what this means is that the cell can no longer divide, and this prevents the cell from increasing in the population. But the consequence is you've lost the ability to repair and regenerate tissue. However, this replicative senescence only occurs in cells that are actually replicating. The majority of cells in the body don't divide anymore, yet they still age. So how do they age? Obviously not through telomere attrition, but through alternative mechanisms. The point being that telomere attrition only accounts for certain cell types in certain contexts, and so it doesn't fully explain the aging phenotype. And so the cells that are likely aging in different ways include neurons, muscle cells, and heart cells. So the last primary cause of aging are epigenetic changes. So epigenetics refers to the information that can be placed on top of the genetic genome sequence. So it can be split down into 3 different categories. So you have DNA methylation, you can have modifications of proteins called histones, and so DNA is wrapped around these histone proteins. And the last category is chromatin remodeling. So chromatin refers to both DNA and histones as a complex. And so the contribution of changes in these 3 different categories can result in a different gene expression patterns. It can result in different DNA repair responses. It can alter the stability of the chromatin. And all of this can affect the function of the cell and hence contribute to aging. But unlike DNA mutations, epigenetic changes are reversible, and that's exciting because if they are reversible, could there be mechanisms or ways that we can alter the epigenetics to reverse the effects that we see in aging? So the important take-home message is that whilst there are many different primary causes, they are all somewhat interconnected, and this can be demonstrated through this diagram here. So during normal biology, inevitable that you accumulate damage in either the proteins, as we saw in proteostasis, or in the DNA with DNA instability, and this can all result in aging. What happens is that this can be advanced through other mechanisms that we'll discuss in the next upcoming videos, such as inflammation and stem cell loss and nutrient signaling that also are important for understanding how these different primary causes contribute to the aging phenotype. So yeah, there you have it. DNA instability, loss of proteostasis, telomere attrition, and epigenetic changes are the primary causes for the aging phenotype.